Alright guys, so last time we left off talking about a protein and we have our long chain of amino acids. So for a protein, we talked about forming dipeptides, we talked about forming polypeptides. And that's when we link our amino acids together, okay? So what happens here is each one of these little guys here, this is called a peptide bond. It's a special type of covalent bond. All right, so in each one, we got our lovely peptide. You guys get it. I ain't gonna go any further than that. Now, on these, we also have different R groups. So I'm gonna say this R group is polar, and let's say this R group right here, this color, is going to be nonpolar. All right, so we're going to do this, 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 okay. Oops. All right, so that's all I need to do there. Here's what I want to show you guys. So what's going to happen here is our R groups, if they are both polar, like these first two here, if this is number 1, this is number 2, these R groups are both polar. So what's going to happen is our R groups are actually going to, if I erase this, they're going to bend a little bit. So they're going to bend like this. And they're going to take their rest of their amino acid with them. But we're going to get a little bond in between these R groups. Now, is it a peptide bond? No. It's actually a much weaker bond, which from last chapter we called a hydrogen bond. Okay, so this is what we meant by the last thing we left off with respect to the hydrogen bonds there. Okay, so what happens here is as our proteins fold, we get our folding due to the polar and non-polar interactions between our amino acids. So look at these two here. These two, the blue ones, they're going to want to come together like this. Okay? So they want to form a hydrogen bond in between. And this red one up here, this one's polar. This one down here is nonpolar, so they want to be as far away from one another as they possibly can because they're opposites. All right, same thing here. These R groups, this red one here, it's going to want to be far away from these guys. All right. Now, once we get enough of these here, guys, the whole thing's going to bend. And that's how we get this gigantic globular state. So, moral of the story with all this example, what causes protein folding? That is going to be the interactions between our R groups. That causes a protein to fold the way it does. All right. So, a while ago, GNC made this protein for people who work out. And it was called Wheyabolic Extreme 60. All right. Wheyabolic is just, it's protein powder, guys. That's all it is. So, what does the 60 mean? The 60 means in one serving, we have 60 grams of protein. Okay, cool. Here's the problem. Your body cannot really synthesize 60 grams of protein at one time. You can synthesize maybe 40, maybe 50. And this stuff's expensive. It's like, I don't know, $70 for one of these things. So, you know, you don't want to waste any. But let's say your body can synthesize 40 grams of protein at one time. If it were a carbohydrate, what happens here is that your body would use some, and then the excess, the part it can't use, the overflow, we would store it in our bodies. Protein's not like that. A lot of diets say eat as much protein as you want. Eat your fill of protein, because protein, your body uses what it can, what it needs, and the rest gets converted, and after that conversion, it gets expelled from your body like that. All right? So difference between our carbohydrates, our lipids, and our proteins. Carbohydrates and lipids, what we don't use, gets stored. Proteins, what we don't use, comes out the other end. Okay, we talked about this last chapter, guys. I just want to kind of focus on it a little bit, just because this is a difficult term, and they always like to put this on the state tests. So when an enzyme loses its shape, you remember we talked about that active site last chapter, it becomes denatured. That's with a change of temperature, change of pH, whatever. Okay, but that's your vocab word there. We have a nice active site on this one. On the other one, when it becomes denatured, temperature gets too hot, pH gets too far out of whack, yeah, loses active site. All right, so here is your picture. Oops, the next slide, sorry. We already talked about enzymes. You guys know they're catalysts just in living things. We talked about enzymes. They can be reused over and over and over and over again. And the enzyme has a direct fit to a part called the substrate, which is like the reactant of our chemical reaction. Okay, here's your picture. So you guys can see we have our enzyme, we have our active site, substrate binds to it. We make the products go away. All right, so yeah. Okay, so we are done with proteins. The only thing we did not do yet, not the right one, here we go. So for proteins, we did our structure, we did our monomer, we did our polymer, we did not do our function yet. So for our function of proteins, we are going to have their building blocks of structures in your body. Okay. They also lower energy needed for reactions. So there is your last one for proteins, and we'll do nucleic acids, and then we will be done with this guy. So take a second, copy that down, guys. All right. Okay, so our last macromolecule, a little different than the other ones because it is not food. Nucleic acids. Their function is to store genetic information. We have 4 different bases we're going to talk about for our nucleic acids. So whatever the monomer is, we have 4 different types for DNA. Here is our structure. So let me draw it out for you guys on our lovely chart here. So we're going to start with nucleic acids. We are going to do our structure of nucleic acids. So let's get it going. So for a structure, we're going to have 3 different parts. We are going to have a circle. That's going to be our phosphate group. Attached to our phosphate, we are going to have, I'm drawing it as a pentagon because it is a 5-carbon sugar. 5-carbon sugar. Now, there's 2 different sugars it could be. For DNA, I'll just put these in parentheses here. For DNA, that 5-carbon sugar is deoxyribose. Deoxyribose. And for RNA, that 5-carbon sugar is called ribose. And then lastly, last part here, we are going to have are nitrogen or nitrogenous. Either word works. Nitrogenous base. And that's either going to be A, T, G, C, or U. Those are your 5 different bases, guys. 4 of them are in DNA. 4 of them are also in RNA. So we're going to have a little bit of overlap, but they stand for adenine is the A, T is thymine, G is guanine, C is cytosine, and U, which is only in RNA, is going to be the uracil. So we'll write that in there too. And so T is only in DNA. The U is only in RNA. All right. So there you go, guys. We are done with the structure. You can either take a picture of that, screenshot it, or if you don't want to screenshot it, you can draw it down. Either way works. So I'll give you guys a second to do that. All right, all right, all right. Cool. Okay. So our 2 main the nucleic acids are DNA and RNA. Our monomer is going to be called a nucleotide, and we already talked about what the structure of a nucleotide is. We put that in our picture. That is our structure. All right. So let's fill out the rest of our chart. We'll get that one done. Oops, I dropped the pen. All right. Okay, so we will do function, monomer, and polymer of nucleic acids. So our function of nucleic acids is going to be to store genetic, you could say material or information, either one of those works. Our monomer is the nucleotide. And again, we have adenine, guanine, cytosine, thymine, and uracil. And then our polymer, guys, we're just going to say DNA or RNA. In reality, these together are called, you don't have to know this, but it's not in your book, polynucleotides, okay? Just a bunch of nucleotides bond together. Yeah, that's all DNA and RNA is. So there you go, and that is done, the end of our chart there. So with that chart, guys, make sure you know all of this chart. Make sure you know if you're going from a nucleotide to DNA, this is a condensation reaction. If we're going from DNA and breaking off some nucleotides, this is going to be a hydrolysis reaction. Make sure you know which one we need water in the reactants. Make sure you know which one we get water out as a product. Okay? All right. All right, so we went over that one. This is the end, guys, of our whole thing on our nucleic acids. I'm going to do one more quick little slide for you here, and then we will call it. So, I want to do some differences between DNA and RNA. DNA has 2 strands. It's double-stranded. RNA is single-stranded. DNA, A, T, G, and C are the nucleotides. RNA, A, U, so that's different. G and C are nucleotides. Okay? DNA, it's found in and never leaves the nucleus. RNA can leave the nucleus. And then our last one here, DNA is much longer. RNA is shorter. So those are your main differences between DNA and RNA, guys. Okay, I just want to give you a few so you can see the difference between them. DNA has 2 strands, RNA only has 1. DNA has thymine, RNA instead of thymine is uracil. DNA is always going to stay in the nucleus. It's never going to leave. For eukaryotic cells, RNA can leave, and DNA is longer than RNA. All right, guys, that is the end of Chapter 3. We will talk about this and start to review for your Chapter 3 test. Have a good day.